Hello everyone, my name is Dr. Bipasha Bose. I work as an associate professor and in charge in the Stem Cells and Regenerative Medicine Center, Yenepoya Research Center, Yenepoya University, Mangalore, India. Now we are going to demonstrate how to detect for the presence of ROS, that is reactive oxygen species, in the live cultures from the mouse ocular surface cells that have been exposed to various doses of ultraviolet-C radiation. The advantage of this technique is that here we can simultaneously detect for the presence of reactive oxygen species live and dead cells without the need to harvest the cells. The principle of this technique essentially lies in the live cell permeability of the dye DCFDA. DCFDA is a live cell permeant colorless dye which during oxidative stress gets acted upon by the intracellular oxidases and gets converted into green fluorescing DCF. Hence, the green fluorescence enables us to detect for the presence of reactive oxygen species in the live cells. On the other hand, propidium iodide is an exclusively dead cell permeant dye which fluoresces red. It is a DNA double-strand intercalating dye. However, the dye Hoechst is permeant to both the live and dead cells. It is a blue-colored nucleus stain. Hence, this is a very simple method in which we can detect for the presence of reactive oxygen species in long-term cultures in a time-dependent manner along with the assessment of dead cells.
Plate 0.2 million cells per 35 mm cell culture dishes. The cells were primary cells from mouse ocular surface. Remove maximum volume of media from each of the cell culture dishes. Leave behind around 500 microliters of cell culture media in close contact with the cells. Minimal